Hello and welcome to the web series, ZeroPM Pieces. In the ZeroPM Pieces, the researchers in the project will tell you about one piece of the ZeroPM puzzle that they're working on. So without further ado, here comes a ZeroPM piece.
All right. Hi, I'm Parviel Chirsir. I'm a doctoral researcher in the University of Luxembourg. And my ZeroPM pieces talk today is going to be about grouping PM substances, cheminformatics, and zebrafish. So what are PMT, VP, VM substances? So these are substances that do not easily degrade in the environment. These are substances that are also easily transported over long distances in environmental matrices, and they are harmful to human health and the environment. So here are examples of PMT compounds, VP-VM compounds, and PMT-VP-VM compounds. To kickstart the ZeroPM project, a list of 38 compounds from 3 structural classes were pre-selected. These compounds were selected based on their substructures, including the triazines, the triazoles, and the PFAS. And these compounds included both parent compounds and transformation products. In ZeroPM, I'm part of work package 5, and my work focuses mainly on transformation products and how these can be used to prioritize PM substances for removal. Now, why are transformation products important? So transformation products are important because when substances are released into the environment, they undergo various transformation reactions to produce different transformation products. Now, generally, these transformation products are more polar and so more mobile than their precursors. But also, some of them can have similar or more persistent and toxic properties compared to their precursors. This means that both PMT compounds and PMT transformation products, or PMTTPs, are a problem. However, there is insufficient transformation product data to be able to assess these PMTTPs. So to give you an idea of how little transformation product data is available on databases, we take, for example, the PubChem database that contains about 122 million compounds, and only about 9,400 of these compounds have transformation product data associated with them. So this is less than 0.01% of the compounds on the database. These transformation products and transformation reactions can be found in a FAIR-TP's dataset. Now, because of the huge number of potential PMT substances, we have to develop a strategy to assess these substances as a group rather than doing one substance at a time assessment. And as part of work package 5, we wrote a reviewed paper where we discussed, amongst other things, grouping strategies to assess and manage PM substances. Now, one of these strategies discussed involved grouping based on commonly retained moieties from transformation products or persistent substructures in transformation products. Now, this is based on the idea that transformation products can have similar persistent, mobile, and toxic properties compared to their precursors. However, assessing these persistent substructures require transformation product data, which have just shown that it's limited. But in this paper, we use cheminformatic tools to try to predict the PM, the persistence and mobility properties of small molecular weight compounds from these 3 substructural groups, so triazines, benzotriazoles, and PFAS. And the prediction results show that substructural group containing melamine, benzotriazoles, and OECD PFAS are predicted to be mobile and non-biodegradable. So a majority of them were mobile and non-biodegradable, showing that these substructures could be persistent in transformation products. Now, to confirm this idea that the cheminformatics results have shown, experimental proof is required. So I am working in collaboration with work package 6 members, including Maxime and Timo, write up a manuscript where we use zebrafish embryos and suspect screening approach to identify these PMT GPs. Actually, the manuscript has already been submitted to Environmental Science Process and Impacts in a theme collection of transformation products and mixtures headed by Hans-Peter and Gabriel Sigmund. Now, the work in this paper is divided into two sections. So the first part involved the use of 5 data poor compounds, selected data poor compounds from triazines, triazoles, and the PFAS group. And I used zebrafish embryos and a suspect screening approach to investigate the biotransformation of these compounds. And to perform suspect screening, I require a suspect list. And for these, I came up with 3 strategies to generate a transformation product data to be used for suspect screening. The first is to use predicted TPs or predicted transformation product data using Biotransformer version 4.0. The second was to download or to get transformation product from literature through PubChem transformations. But since this case I selected data poor compounds, you only had 1 transformation product from chlorsulfuron. And then another strategy was to come up with analogous transformation products. So these are transformation products that I drew from structurally similar compounds to the precursors of interest that have transformation product data in databases. So I'll walk you through the analogous TP generation workflow. So first, I take the parent compound of interest. So in this case, symmetrine. I do a similarity search on PubChem to get the structurally similar precursors. And then I filter for these compounds that have transformation products. Now, this leads me to compounds like terbutrin, ametrine, and prometrine that have unique transformation products associated to them or with them. And then I download these transformation products and then observe and note the transformation pattern. So for example, terbutrin goes to D-ethylterbutrin through an N-dealkylation, which means it loses an ethyl group here. And you can see the transformation product has also lost an ethyl group. So I draw a similar transformation reaction for symmetrine that has no transformation product in the database. And then I do a loss of an ethyl group. So this ethyl group is lost, and to come up with this ethyl symmetrine. So this ethyl symmetrine in this case is what is called an analogous transformation product. So I do this for all the other transformation reactions and for all the other data poor compounds to come up with a suspect list that I use for suspect screening. For the second part, we had 36 PM compounds from Carlier et al. Study. So Maxime Carlier, who is also present in this call, he investigated some of these substances. And these substances were based on the triazines, the triazoles, and the PFAS subgroups. Since some of these compounds already had transformation products on PubChem database, I basically predicted the transformation products for these and downloaded the already available literature transformation products. The next part was to perform the zebrafish exposure experiments. So why I use zebrafish as in this case is because zebrafish are environmentally relevant species. So they are freshwater organisms, and also they grow quite fast and have an early established metabolism. And also, zebrafish share genetic and physiological similarity with humans, so the results are translatable. Now, for the exposure experiments were performed in 2 parts. So the first part was performed in LCSB by me, and the second part by Maxime. And for the first part, I exposed the 5 data poor compounds individually in 6 well plates to zebrafish embryos for a duration of 72 hours. I also had untreated controls where there was just the zebrafish embryos without the compounds. I, of course, had the case with the zebrafish embryos and the compounds. And then I had stability controls that only had the compounds without zebrafish embryos. Then the second part performed by Maxime, he exposed these 36 compounds individually in 24 well plates for a duration of 92 hours. And in the end, he sent the results over to LCSB, to me, for further analysis. Now, once all the exposure experiments were done, they were all saved in the -80 freezer until extraction. And extraction, in this case, involved first homogenizing the samples with ceramic beads, chloroform, and methanol, and then transferring the supernatant for liquid-liquid extraction with chloroform and water. And then the lower phase that contains the non-polar compounds were then dried. The samples were reconstituted and filtered, and then reverse phase liquid chromatography high resolution mass spectrometry was used to measure these samples, to measure the analytes in these samples. Now, for the mass spectrometry measurements, I used the Q-Exactive HF-OB trap. And once the measurements were done, I had the raw files, and then I converted them to .mzml files, which is a compatible format for the data mining software that I used. And for this data mining, I used Patron, which is an R-based open source software that integrates other cheminformatic tools such as Medfrac for in silico fragmentation and MassBank for spectral annotation. And with these, I could perform the suspect screening using the two separate suspect lists for the different parts of the experiments. And then I could identify the parent compounds and the transformation products in the samples. So some of the results include for the data poor compounds, in total, 33 transformation products were annotated from 4 precursors. So no transformation product was annotated for Adona. And the highest number of TPs were annotated from Symetrin, followed by clossiferon and metconazole. And the annotated TPs were from the different strategies for TP generation. So the highest number in this case was from analogous TPs. And we also had TPs annotated that were both from analogously drawn and from biotransformer predicted TPs. And then we also had TPs from biotransformer predicted TPs. Now, these results show that analogous TPs are a good source of transformation product data when structurally similar compounds have transformation product information. Now, for the second part, using the 36PM compounds, in total, 56 TPs were identified from 20 precursor or parent compounds. And the majority of these annotated TPs were from the triazoles, followed by the triazines, and then PFAS. Now, in this case, the highest number of annotated TPs were from biotransformer predictions, and followed by those that were both from biotransformer predicted and Ligitor TPs. And then we had Ligitor TPs annotations only. Now, this result also show that predicted transformation products are a complementary source of transformation product data to Ligitor transformation product or non-TP data. Now, the TPs annotated showed evidence of the 2 metabolic pathways in zebrafish embryos. So we had phase 1 metabolic pathway, that metabolic transformation reactions such as N-dealkylations. We also had others such as thioether substitution. But then we also had phase 2 metabolic reactions such as glucuronidation. But what was also interesting is that we found some transformation products or some TPs also in the stability controls that did not contain the zebrafish embryos, showing that these compound, Symetrin and the others that had TPs identified in the stability controls, could also break down on their own. So the transformation could be both biotic and abiotic transformation. So here is an overview of the transformation pathway of an example parent compound, so Symetrin in this case. You can also notice other metabolic transformation reactions. So we also have other phase 1 reactions like oxidation. We also have S-demethylation. But you also notice here that the 135 triazine ring marked in red is retained in all the transformation products. Now, this is a similar case for all the triazine-containing compounds. So one could say that the 135 triazine ring is a persistent substructure in the transformation products of triazine-containing compounds using the zebrafish embryos. Now, for benzotriazoles, we also had a similar case of phase 1 transformation reactions and phase 2, but also the benzotriazole ring was also conserved in these transformation products, showing that the benzotriazole ring is also a persistent substructure in transformation products. A similar case is was also noticed for the 124 triazole ring, which was also conserved in the transformation products, showing that this is also a persistent substructure in transformation products. And for the PFAS, we had CF3 and the CF3 and the CF2 groups also conserved in transformation products, showing that these groups are also persistent subgroups or persistent moieties in transformation products. What was also interesting about PFAS is that we found some amino acid conjugated transformation products, such as the glycine and taurine conjugations. This has also been previously reported in literature, showing that these could be in important routes for environmental contamination for PFAS. Now, we also found some cases, for example, where both the parent compounds and the transformation product were of concern, like this PFBS, which is a transformation product of PFHXS, so perfluorohexane sulfonic acid. And these two have been registered in the candidate list of substances of very high concern, showing that both of them are of health or environmental concern. Now, to summarize the results of this study, in total, I generated 90 transformation products. 56 of these TPs were not yet associated to parent compounds or have not been associated as transformation product data in FAIR TPs. And 29 of these structures were not in the PubChem database. And we generated mass spectral data from standards of 15 compounds. So these results could help increase transformation product annotation, identification, and risk assessment. Now, a few take-home messages from here are a multi-source TP suspect list increases transformation product coverage and annotation compared to using a single source transformation product list. The 135 triazine ring, the 124 triazole, the benzotriazole rings, and the CF3 and CF2 groups are persistent substructures in transformation product of their corresponding parents. And then these persistent substructures can serve as markers or indicators of persistent transformation products. Now, I'd like to end my presentation by giving a huge thanks to my supervisor, Professor Emma Szymanski, and also to my Work Package 6 collaborators and co-authors of the PMT TP's paper, so Lorena, Maxime, and Timo. And also a huge shout out to the Work Package 5 members for the grouping paper. And a huge thanks to Environmental Cheminformatics Group, especially Emma Palm and Camila. And of course, major thanks to ZeroPM for the funding and giving me the opportunity to do this work. So thank you very much. And if you have any questions, don't hesitate to ask.
ZeroPM, zero pollution of persistent and mobile substances. This project has received funding from the European Union's Horizon 2020 Research and Innovation Programme under grant agreement number 101036756.